Hello, in today's video, we shall learn in detail about Selaginella, which is a pteridophyte. By the end of this video, you'll have a clear understanding about the morphology of Selaginella, anatomy of its stem, root, rhizophore and leaf, mode of reproduction both asexual and sexual, its gametophytic generation, fertilization, embryo development, and finally its life cycle. Let us begin with the morphology of Selaginella. Selaginella is a pteridophyte commonly known as spike moss or club moss. They are evergreen, delicate, non-flowering, seedless plants, mainly growing in tropical regions. Selaginella are mostly herbaceous perennials with dorsoventral shoot. The stems may be aerial, horizontally creeping on the substratum, as in Selaginella cruciana, commonly known as the golden club moss, or maybe suberect, as in Selaginella trachyphylla, or erect, as in Selaginella erythropus. Under dry conditions, some xerophytic species like Selaginella lepidophylla, Selaginella pilifera can survive dehydration. During dry season, the plant rolls up into a compact brown ball and can unroll, become green, and carry on normal activities when exposed to water. This phenomenon is known as poikilohydry, and the plants are known as poikilohydric plants or resurrection plants. The main plant body is a diploid sporophyte and is well differentiated into roots, stem and leaves. The plant is evergreen, profusely branched, delicate herb with dorsoventral shoot, radial creeping branches, small scale-like leaves called microsporophylls found on the stems from which roots arise. Numerous small, simple leaves with pointed apex are arranged spirally on the stems and branches. The leaves possess a single unbranched mid-vein. Leaves in subgenus Homeophyllum are of same size, spirally arranged, forming a dense covering. Leaves in subgenus Heterophyllum are dimorphic, that is, they are of two sizes, small and big, arranged in pairs. On the adaxial side of the leaf, near the base, is present a small membranous outgrowth called the ligule. It is made up of two parts, glossopodium and the body. Glossopodium is the basal hemispherical part made up of large thin-walled cells and is surrounded by a glossopodial sheath. Above this is the body made up of many cells. Colorless, elongated, cylindrical appendages known as rhizophore develop from the prostrate axis of the stem at the point of dichotomy and grow downward into the soil and give rise to a tuft of adventitious roots as the dichotomously branched primary roots are short-lived. Sporophylls or spore-bearing leaves are present in clusters known as strobili at the tip of the branches and bear sporangia. The plants are heterosporous with spores of different sizes. The larger ones are megaspores and the smaller ones are microspores. The gametophytic prothallus is heterothallic or dioecious. The plant shows haplodiplontic life cycle in which the sporophyte is the dominant phase in its life cycle. By the way, if you're getting some value out of this video, please like and share this video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now, let us understand the anatomy of Selaginella stem. Transverse section of Selaginella stem shows an outer epidermis, middle cortex, and a central stele. The outer epidermis is single-layered, made up of parenchymatous cells, and is protected by a covering called cuticle. Stomata is absent. The cortex is well defined. It is either parenchymatous or differentiated into outer sclerenchymatous hypodermis and inner thin-walled parenchymatous region. Intercellular spaces are generally absent. There are radial elongated cells called trabeculae by means of which steles are attached to the cortex. The trabeculae cells have Casparian strips. Air cavities are present in between the cells. The stele is surrounded by a single layered pericycle made up of parenchymatous cells. The xylem may be monarch, diarch, or multi-arch. Typically, stele is protostelic with xylem in the center surrounded by phloem. Pith is absent. Xylem is usually made of tracheids. Vessels are completely absent. Xylem is exarch with one or two protoxylem groups. Phloem consists of sieve cells and phloem parenchyma but lacks companion cells. Next, let us learn about the anatomy of Selaginella root. Anatomically, the root consists of centrally located stele surrounded by cortex, epidermis, and a thin cuticle. Transverse section of Selaginella root shows an outer thin layer of cuticle covering the one cell layer thick epidermis. Few of the epidermal cells give rise to root hairs. Below the epidermis are a few layers of parenchymatous cortex. Sclerenchymatous cells may be seen in old roots. Endodermis is not well developed. 1 to 3 layered pericycle is present. Stele is protostelic, monarch, and exarch. That is, protoxylem is found towards the periphery. Now, let us look into the anatomy of Selaginella rhizophore. Transverse section of Selaginella rhizophore shows a thick-walled epidermis which lacks stomata and root hair. It is covered with a thick cuticle. A thick-walled sclerenchymatous hypodermis is present, followed by parenchymatous cortex. Endodermis is not well developed. A single layered parenchymatous pericycle is present. Stele is protostelic, monarch, and exarch like the roots. Next is the anatomy of Selaginella leaf. Transverse section of Selaginella leaf shows an upper and lower epidermis made up of parenchymatous cells containing chloroplast. Stomata is present in the lower epidermis. The mesophyll cells are not differentiated into palisade and spongy cells. They are made up of parenchymatous cells with intercellular spaces. Single vascular bundle is present in the center and is surrounded by a bundle sheath. The median vascular bundle contains xylem tracheids, which are surrounded by sieve elements of the phloem. Next, let us study the various modes of reproduction in Selaginella. Selaginella reproduces both asexually as well as sexually. Asexual reproduction takes place through fragmentation, tubers, or through resting buds. Sexual reproduction is by means of spores. Asexual reproduction by fragmentation. Under humid conditions, certain species of Selaginella, like Selaginella rupestris, develop adventitious branches on the trailing branches of the stem. These adventitious branches separate from the parent branch and develop into individual plants. Asexual reproduction by tubers. Tubers develop towards the end of the growing season. They may be aerial or subterranean. During favorable condition, they germinate into a new plant. Asexual reproduction by resting buds or dominant buds. The leaves at the apical end of some aerial branches are closely arranged and cover the growing points. During unfavorable conditions, they behave as dormant or resting buds. Under favorable conditions, they become active and form the rhizophores that bear roots at their tips. Next, let us learn about sexual reproduction in Selaginella. Selaginella is a sporophytic plant and reproduces sexually. Strobilus is a reproductive structure formed at the apex of branches. It consists of a central axis and spirally arranged, dense, fertile leaves known as megasporophylls and microsporophylls. Stalked, lobed megasporangia develop on the megasporophyll and give rise to 4 megaspores. Stalked globular or elongated microsporangia develop on the microsporophyll and give rise to large number of microspores. The plant is therefore heterosporous. Megaspores form the female gametophyte, while microspores form the male gametophyte. Next is microsporangium and megasporangium. Each microsporangium and megasporangium is stalked with two-layered thick wall followed by a single layered tapetum. Both the types of sporangia consist of a mass of sporogenous cells. In microsporangium, these cells form the microspore mother cell, and in megasporangium, they form megaspore mother cell. The microspore mother cells undergo meiosis and form numerous small haploid tetrads of microspores. All the megaspore mother cells except one degenerate. The one that survives undergoes meiosis and produces a tetrad of 4 large haploid megaspores. A vertical slit on the apical region of the sporangia helps the spores to be liberated and are carried by wind. Now, let us look into the gametophytic generation of Selaginella. The microspores are small, unicellular, uninucleate structures and are surrounded by an outer spiny thick wall known as exospore or exosporium or exine, and an inner thin wall known as endospore or endosporium or intine. On germination, the microspores give rise to the male prothalli. The megaspores are large and are tetrahedral in shape with a triradiate ridge. The spores are covered by 3 wall layers, namely outer exospore, middle mesospore, and inner endospore. The megaspores on germination give rise to the female prothalli. The development of the male gametophyte takes place in situ, that is, it starts within the microsporangium. The microspore divides forming 2 unequal cells. The smaller is the prothallial cell, which does not divide further, and the larger is the antheridial cell. The antheridial cell divides to form 2 primary cells of the antheridium. These primary cells undergo divisions to form 4 primary androgonial cells and 8 jacket cells. So, a 13-celled gametophyte is formed. It consists of 1 prothallial cell, 4 androgonial cells, and 8 jacket cells. At this stage, the exosporium ruptures and the spores are liberated. The primary androgonial cells divide repeatedly to form 128 or 256 androcytes or anthrozoid mother cells. Each anthrozoid mother cell forms a single anthrozoid, which is a spiral, uninucleate, biflagellate structure. The 2 flagella are of unequal size. The endosporium ruptures and the anthrozoids swim in water and find their way to the neck of the archegonium. The development of the female gametophyte takes place in situ, that is, when the megaspore is still inside megasporangium. As the megaspore increases in size, a large central vacuole is formed. Free nuclear divisions take place and later number of nuclei increase in the apical region. It is followed by wall formation, due to which an upper cellular region known as female prothallus and a lower noncellular region known as storage region are clearly visible. The wall of the lower cells become thick, forming a diaphragm. With the increase in cytoplasm content, the vacuole disappears. At this stage, the female gametophyte is liberated. When it falls on a suitable substratum, it germinates. The walls rupture, the cellular tissue comes out and forms rhizoids. These rhizoids help the gametophyte to fix to the substratum. Next is the development of archegonium. In the female prothallus, few cells form the archegonial initials and give rise to archegonia. The archegonial initial divides transversely to form an upper primary cover cell which forms the neck by undergoing vertical divisions at right angles to each other, and a lower central cell which divides to form an upper primary neck canal cell and a lower primary venter cell. The primary neck canal cell forms a single neck canal cell. The primary venter cell divides to form a ventral canal cell and an egg. Embedded in the female gametophytic tissue is the archegonium, which is a flower-shaped structure. Each archegonium consists of a short neck and a broad venter. At maturity, the neck canal cell and the ventral canal cell disorganize. They create a pressure by absorbing water. The cover cells get separated and the anthrozoids successfully enter the archegonium and reach the egg. Once the anthrozoid reaches the egg, its nucleus fuses with the egg nucleus to form a zygote. This zygote secretes a protective wall around itself and is known as oospore. Parthenogenesis is seen in Selaginella intermediae. The egg develops into embryo without fertilization. Next, we shall look into the development of embryo. The oospore divides transversely, forming an upper suspensor initial epibasal and a lower embryo initial hypobasal. The upper suspensor initial divides to form a multicellular suspensor which helps the developing embryo to get nourishment from the female gametophytic tissue. The lower embryo initial divides to form four-celled embryo. A shoot initial is formed along with a multicellular structure which differentiates into foot, rhizophore, stem and cotyledons. Finally, let us summarize the life cycle of Selaginella. In Selaginella, the sporophytic and gametophytic generations alternate with each other. Selaginella is a diploid sporophytic plant. The plants are heterosporous, producing microspores and megaspores. The former produces male haploid gametophyte and the latter produces the female haploid gametophyte. They grow upon the strobilus and produce anthrozoids and egg in Antheridia and Archegonia respectively. These reproductive structures after fertilization produce zygote, which on germination gives rise to a sporophytic plant. Thus, the sporophytic phase is dominant over the gametophytic phase. So today, we have learnt in detail about morphology of Selaginella, anatomy of its stem, root, rhizophore, leaf, its asexual and sexual mode of reproduction, and also its life cycle. I have some practice questions for you. Please share your answers in the comments section below. You can pause the video and write down the questions if you like. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comments section below. I'll see you there. Goodbye. All the best.